We're very lucky in this state to have Melbourne University professors, associate professor, PhD students so close by, and we can hear straight from them what kind of work they're doing. There's something for everybody here in the three talents that we're going to hear from. Professor Rudi Appels is an honorary professor in wheat genetics at the Uni of Melbourne. He's been awarded the Farrer Medal in 2022 for his work understanding the structure and function of the wheat genome. William Farrer was one of the first wheat breeders and probably the first wheat breeder in Australia back in the very late 1880s, 90s and into, I think he finished up in 1923 or...
He passed away in 1905.
1905, okay, I've gone way beyond. Rudi's appointment at the uni allows him to work with research colleagues, postdocs and graduate researchers to understand biochemistry of agriculture and food production. His research papers have covered all kinds of subjects like tracking drought response in wheat heads, as well as working in dough systems and looking at water absorption and microbiology. So that's a wonderful array of topics. Anna Wittwer is a PhD candidate at the Melbourne University, working with Associate Professor Kate Howell. They're studying the properties of lesser known yeast in sourdoughs in an attempt to modify the nutritional quality of bread. Anna is leaving for France very soon to spend 4 months with Delphine Sicard at her laboratory in Montpellier. Delphine Sicard was one of the keynotes at the 2019 GrAiNZ festival. She's a fantastic French resource on sourdough microbiology and biodiversity. So thanks for sharing, Anna. And then our friend Kate Howell, one of the organisers of GrAiNZ and all the panels, she's an associate professor now in the food chemistry lab, or School of Food Chemistry. She leads a laboratory of postdoctorates and graduate researchers to investigate microbiological ecology and chemistry in food production. And she's passionate about understanding fermentation for flavoursome, delicious and nutritious foods.
Okay, let's hop into it. So I'm Rudi Appels, and I've got this honorary position in Kate Howell's group. So we sort of try and work together. I come from a very basic side of things, and then, but I really enjoy working with the more applied areas. And just in case I can catch anyone afterwards, I've been doing some work on the vortex milling with Axel Mills in Bayswater, which has been quite informative. Okay, so what I'd like to share with you is what is happening in the genetics of the crops. Wheat will be what I focus on because that's what I do, but in fact I'll finish up to indicate that it's actually all the crops that we deal with, the chickpea, you name it. We now have an understanding of the genetics and the capacity to trace traits of interest that we've just never had before. And that's only in the last decade, or the last 5 years even, for that to happen. Okay, grain diversity is the real focus at the basic end of things. And I'll explain that as I go. Okay, so Kate asked me to talk about the family tree, so we'll start at the beginning. We have the Middle East is where a lot of many of the crops that we use in our systems originated, and we have the einkorn wheat, emmer wheat, barley, lentils, peas, and of course the chickpeas as well. And it's about I'll go to the next slide. About 10,000, roughly 10,000 years with respect to wheat that we saw we can find paleontological evidence that the hexaploid wheat was being grown when people have excavated ovens and looked at the residues that was there. So we have... is there a pointer on this? No. So we have the early period, where the diploids, the two diploids, what we call the A genome, what we now know as einkorn wheat, crossing with a B genome species, and they form what we now know as the durum wheats down here, and that crossed in that area in the red. In the field there was a random cross, and it's actually quite an easy cross to do between the durum wheat and the goat grass, which has become very popular in recent years, as I'll come to in a minute. Then there was a period where it's been taken a while to work out. It wasn't just a straight lineage to our current wheat, which we thresh and get the grain. There was a crossing, intercrossing, and for quite a long time the spelt was quite popular and was grown. It's got obviously not that easy to thresh, and it's been that sort of mixture, but eventually the societies selected for the threshing. It's interesting, about 2,000 years ago, there are reports in the Chinese literature of spring and winter wheats being recognised. Okay, so with part of the family tree in terms of the Australian aspect, we have William Farrer, who was really the starting point for us. His line of federation was released at the turn of the century in 1900s, and with the genomic, the detailed DNA analysis that we can do now, which is really incredible. We can go back through his pedigrees and the material, the pups. He started with purple straw and the Etowah and the improved 5. So he was really, the pioneering part of what he did was to actually communicate with his colleagues at the time by mail and got samples from all over the world, because intuitively he felt that diversity was the answer to really try and increase both resistance and tolerance and drought tolerance in the material that was being grown in New South Wales at the time. So the interesting thing is the greenish dots that I've outlined, we've actually had this interest in a gene that gives you the udon noodle quality, the Japanese udon noodle quality. And we can actually trace that through insignia, and then, of course, there's all the current ones. We can trace that back to material that Farrer actually introduced. So although he didn't know it, he really set the basis for what has been an incredibly good industry for the West Australians in terms of exporting udon noodles, quality wheat to Japan. So that's been one of his sort of a real legacy that he left us, which has been valuable. But his basic thing was to do wide crosses, and that was to get this increase in diversity in the material that he could select. And federation was the result. That period of Farrer, there was a lot of activity in Europe. Strumpelli was a particularly innovative breeder. He introduced the dwarfing genes, the short stem, quite a few decades before the big program of Borlaug in that area. So Strumpelli introduced a dwarfing gene from the Japanese wheats, and he produced material which was really the basis for a lot of the European winter wheats for quite a long time. And then we come to the 1960s, where we have Norman Borlaug and his really exciting worldwide based effort for crossings and testing based at Cimet, Mexico. both of these achievements, the key was to reduce the stem length and get more of the nutrition and effort of the plant to go into the grain. And CIMMYT, the program that was and is still based in Mexico, is still really active. So in the 40s and basically up to the current date, the CIMMYT program, I'm just picking a few highlights for you here to share with you. The CIMMYT program really introduced a lot of what we call synthetic wheats. So I was involved in that back in that time, and they produced the big collections that had been collected by a collector from Japan who'd gone to the Middle East and collected all the D genome, the goat grass, you know, that spindly little wheat head. But in fact, and that's in fact the least of the variables in the current wheats. And what they've done is to then say, okay, well, let's just cross the accessions of the cultivars, the D genomes into the hexaploids and reproduce what nature did at the turn of in the 10,000 years ago. And that's been a huge program. 1,500 synthetic wheats now carry this, what's basically called the modified D genome. But basically it's introducing more diversity for selection in the different environments that are around the world, and for quality as well. And so to date there are 80 of these wheat varieties, these synthetic wheats, are being released worldwide. So it was a big program and a huge crossing, which you could only do in a place like CIMMYT, Mexico, and they've really are proving to be very successful. Okay, so complementing that in terms of the, if you like, the wheat family, the tree, and also all the other crops. There were, during this period of just using that timeline, you have advances in looking after the soil, precision agriculture, and management issues. And as we've just heard this morning, the storage of the grain, which is something I'm particularly keen on as well. Okay, so present day. We have, this is just a snapshot of what's happening with nerdies like me interested in the DNA of the crops. You can produce, this is a pictorial image of fingerprints for the whole genome. So what's very powerful, these are, for your information, these different colours are just indicating different single base changes that are differing between the reference genome, which is called Chinese Spring. And so when it's all down below the maize and Westonia, they're basically hardly any different from the reference genome. And then Lancer and Shuyan are very different. The bottom line is you can identify these fingerprints for regions. So for a long time, we were sort of trying to use single genes, if you like, to track the complex traits that everyone in the farm industry knows that are sort of complex to deal with. Here we can actually look at regions and actually track phenotype, fingerprints to track the phenotypes. And just to give you a reality check here, in Australia we do these large large national trials, and we looked at the group in AgriBio here in Victoria looked at almost 700 lines doing this. So this is already now small scale. We can do it at a much larger scale. And they found that there are two major fingerprints, which are H1 and H2, which I've indicated up there, which are in this region where I reckoned there was a gene that was affecting the yield enormously. And in fact, that whole region where this gene is, the one I've indicated up there, there was over this trial, which is in the field, it's all sort of dealing with real life situations. the H2 phenotype, which is the one that's the two down below, was clearly had a big advantage, and in terms of what breeders have been selecting in their extensive trials, and then it turned up in these national trials. The interesting thing out of this, there's an H3 phenotype, the one in grey there, which actually out-yielded the H1 and H2, but it hasn't been really selected for. There was probably some associated defects associated with that phenotype. So this is just to sort of give you an idea of what's happening. And in Victoria, you've got here the Department of Agriculture. Matt Hayden's running in charge of that group, where they've got the big grant from the GRDC, which is now looking at the entire Australian grain collection in Horsham at this level. So you can actually just start to really do some. If you find a particular trait in one of the wild germplasm they have, you can then do the fingerprint analysis and track it into your system of interest. And this applies both to yield and to grain quality, which is more an area that I'm interested in, and nutrition, which has already been mentioned here. Okay, so just to let you know that I'm not just on wheat. So I went into Matt's office and asked him, can you give me an example of chickpea for this meeting? And it turns out that this whole technology is now giving is being used by commercial companies to really select for genetic diversity. Again, it's going back to that out of the wild germplasm, which is giving the high protein levels. And there's also big programs. This is a really challenging disease in chickpea, the ascochytid. And it's, again, in the wild germplasm. Nature's done it all before. We're limited in what we can do with genetic engineering because society doesn't want us to fiddle in the lab with our crops. But in fact, nature's done it all before. So if we know what to look for now, particularly with these DNA fingerprints, we can get the material prepared. So just to finish up and basically lead into what Anna's going to talk about, this is from the wheat side of things. If you're inspired to get a low FODMAP wheat, the region of the chromosome has actually got 8 of these genes coding for the fructan levels in the grain. And so it's not just a simple matter of just going in and mutagenizing, because if one's gone, the other 7 will kind of compensate. And so there's a real challenge there of using, again, natural variation, which has got either the whole locus missing or different numbers missing, and doing a concerted effort to produce wheats, which you can then sort of look at from it can see if it can complement what you'll hear from Anna. As I say, genetic engineering, you could do this by genetic engineering very simply, but the product just isn't accepted to society. So the DNA fingerprint technologies could now be designed to really select for these changes and get the varying numbers of genes. Okay, so I guess we'll have questions and discussion afterwards. Yep. So I'll hand over to Anna.
Hi everyone. I might just adjust that slightly. As Emily mentioned, I'm Anna. I'm a PhD student in Kate's lab. And today I'm going to be talking about fructans and some fun sourdough yeasts. So before I talk about the what of my presentation, I thought I'd talk a little bit about why I'm interested in this stuff. And it starts when I was a teenager. My older brother was diagnosed with Crohn's disease, which is a subset of irritable bowel syndrome, or IBS. And for people with IBS, some nutrients are not well absorbed in the gut, and there's other things as well, but that leads to some fairly unpleasant gastrointestinal symptoms. And IBS affects about 10% of the world's population, which is a lot of people, so it's not an insignificant problem. One of the main interventions for things like IBS is they change your diet. So they say, don't eat this, do eat this. And my older brother was given a really long list of things that he could no longer eat. And one of them was onion and garlic. And I remember thinking as a teenager, oh, you know, it sucks for you, but I don't see how this is going to affect me. But of course, he still lived at home, so family meals were still a thing. And if you've ever tried to cook a family dinner without, well, any meal without onion and garlic, it's very hard. So the problem with onion and garlic for my brother is that it contains this carbohydrate called inulin. And inulin is actually part of an even bigger gang called the FODMAPs, which stands for fermentable oligosaccharides, disaccharides, monosaccharides, and polyols. And they are this group of dietary carbohydrates that trigger IBS symptoms in some people. And they're really common. They're in a lot of really common foods that you eat. So lots of dairy, some fruits, honey, pulses, grains, a lot of very healthy foods, really. So my brother is FODMAP intolerant. He can't have any of these, but he can still have bread, thank God. But he can only have one kind of bread, and it's only this Edwards spelt sourdough. He loves it. It's his favourite thing on the planet. So why this bread? The first is that spelt GrAiNZ, spelt flour, is lower in total FODMAPs than rye and wheat flour. And the other part of the equation with this bread is that it's a sourdough. And the sourdough fermentation pushes the FODMAP content even lower, so much so that this can be considered a low FODMAP product. So why, I guess, why spelt and not other things? The answer is fructans are the main FODMAP that's found in bread. They're in the blue. Depending on the type of flour that you use or the type of bread it is, they are typically the largest contributor to FODMAP content. There are others, as you can see here, there are other fructans, but, FODMAPs. Both start with F, it's too hard. There are other FODMAPs, but for IBS people, fructans are the main obstacle to eating bread. This is what they look like. Oh, pardon me. You can see that wheat fructans and inulin look very similar. Well, I guess they are very similar. The main difference is that inulin is usually one big, long, straight chain, whereas fructans are very branched, so they tend to be more like a tree relative to compared to a stick. So with these kinds of products, so we've got spelt sourdough, and then there's also this low FODMAP flour. You could consider this job done, and for my brother it kind of is. The problem with these solutions is that they're very expensive, especially the low FODMAP flour. And that's hopefully where sourdough yeasts come in, to maybe offer an alternative that might not be any better, but it would be an alternative, and it could maybe be cheaper, which would be good. This graph shows us the FODMAP composition of different white wheat breads made with different techniques. So we've got a ciabatta, a standard bread, and then we've got sourdough. And what you can see is that the FODMAP profile of the sourdough bread is quite different from the FODMAP profile of the other two styles of bread. And notably that the fructans in green are much lower in the sourdough bread than they are in the other two types. We assume that this is because of the way sourdough is made. And we can categorise some of the differences between sourdough bread making and standard bread making into sort of some broad categories. And one of those categories relates essentially to how long we let bread ferment for. If you look at it from a certain direction, you can consider the part of a sourdough starter that's added to dough to make it to be basically like really, really pre-fermented dough. And the other thing is that sourdough bread tends to be proofed for longer. Not always, but it tends to be proofed for longer than standard bread. So all of that adds up to us letting bread ferment for longer. And we already know that just extending proofing time in standard bread allows us to lower dough fructan content. So in this case, this group of researchers, they fermented something for 4.5 hours, and you can see that the fructans in the green go down over time. And the interesting thing is that they also found that was true regardless of whether you use bread wheat or spelt. Obviously the spelt starts at a lower point, but there's still a decrease. And we think, well, the most logical assumption here is that what we're doing is giving the fermenting microbes a longer window in which to make changes to the dough. That's not the whole picture, though. It's not just that we're giving the yeasts more time. It's that in sourdough, it's a different cohort of microorganisms altogether. When you would make a standard bread, you'd use a pure industrial Saccharomyces cerevisiae strain, whereas sourdough bread tends to contain 1 or 2 yeast species and 1 to several bacterial species. You'll notice that Saccharomyces is on the list of sourdough yeast species, and they do naturally occur there, but we've also got some other cool, fun, exotic-looking names, as well as a bunch of lactic acid bacteria whose contribution shouldn't be understated. We know all of this because there's quite a body of research into sourdoughs worldwide. This is a map of the world with every country coloured by how many types of sourdough products it has, and Italy is by far and away the sourdough kings of the world. They have 71 types of sourdough products. I think it's because they have a lot of sweet sourdough-based things. But this also serves as kind of a reflection of the global interest and work that's being done in sourdoughs. You can see that there's quite a bit of activity in Europe, some in Asia and North America, but Australia remains stubbornly purple. So the contribution, our small contribution to maybe filling that gap, is that students and researchers like me from Kate's lab, we've been looking at sourdoughs from Victoria and southeastern Australia for a little while. There's quite a lot of us down in the corner. We've looked at 35 sourdough starters. And we usually get, as I mentioned before, we usually only get 1 yeast, sometimes 2 out of a starter. And all of the species that we've got are on the right in the green. As you can see, we have a lot of Saccharomyces cerevisiae. We find that a lot in sourdoughs, and we also have quite a few Kazakhstania and Pekia yeasts. The starters that we have in red are ones that we've actually made ourselves in the lab from a variety of different grains. And we've tested quite a few. We've gone from sort of varieties of wheat all the way to things like brown rice and millet and teff. But the ones in blue are ones that we've taken from bakeries and starters sourced from across Victoria, which you can see on this map. There's not really a pattern to which species are found where. If there is a pattern, we would need so much more data to see it, I think. But you can see that there's quite a concentration of spots over Melbourne just because that's where we are. But yeah, it's just really cool to see. What I'm interested in is whether maybe we can use some of these interesting Victorian sourdough yeasts to make low FODMAP bread. There's already work being done on using yeast to lower the FODMAP content of bread elsewhere. This is the work of a group of Belgian researchers who have looked at this yeast called Cluveromyces marxianus. And what they found was that over 3 hours, this yeast is able to reduce, or it's able to get rid of over 90% of the fructan content in wholemeal flour. You can see it in the blue on the right. Which is quite something because wholemeal flour is higher fructan than white flour. This yeast is special because it produces an enzyme called inulinase, which, as the name suggests, is very good at breaking down inulin, and by extension, similar molecules, if you remember, like fructans. And it sort of breaks them down into their subunits. There's another group of researchers in Ireland who are looking at a different yeast called Lachantia fermentati. So we've got all of these different bread FODMAPs in the different panels, and Lachantia is up the top and baker's yeast is down the bottom. That's their comparison. The cool result, obviously, here is the fructan panel in the top right. The bread made with Lachantia fermentati is about half the fructan of baker's yeast bread. And as an extra bonus, they also found that the bread they made with this yeast was able to meet established low FODMAP cutoff values. So that's a very special one. I'm not interested in either of those because I'm going to be different. I ignore all of the green and red writing. I'm interested in a group of yeasts called Kazakhstania that's sort of a distant cousin of Lachantia and Cluveromyces. You can see it up there in the orange. It is named after Kazakhstan. That's usually the first question I get about it. The first yeast from that genus was isolated there. And among the sourdough yeasts, it's actually quite a big deal for a obscure sounding yeast. From sourdoughs worldwide, 7 different species have been isolated, which actually makes it the best represented yeast genus in sourdough. So we know that sourdough is low in fructans or lower in fructans. We know that Kazakhstania is a very common sourdough yeast and that it has some special relatives. But no one's considered whether it might be a good tool for reducing the fructan content of bread. So that's what I'm interested in looking at. The first thing that we did here is look at a big family tree of this genus. So this is every species in the genus. There are about 52. And we've just indicated the ones that are associated with sourdoughs or breads with a red spot and the ones that are not with the blue triangle. And what we noticed is that the Kazakhstania yeasts associated with sourdoughs, not all of them, but most of them, tend to cluster together in the middle in this little clade. And that maybe tells us that they have some shared genetic character that maybe is part of why they're good sourdough yeasts. And that shared genetic character could be useful to me. We did something similar, but this time not looking at the yeasts themselves, but at the enzymes they make. So this is the sugar breakdown enzymes of a bunch of bread yeasts. And we wanted to see not just, I guess, how similar the yeasts themselves were, but how similar the tools they have were. And again, we found that the enzymes that Kazakhstania yeasts produced are most similar to other Kazakhstania enzymes. So again, that suggests that there's something about these little machines that is similar and maybe unique to this genus. So now we test that. We have a collection of 29 Kazakhstania yeasts, two different species, and for these tests we also had included some baker's yeast and some Cluveromyces marxianus as positive controls. To do that, we made some agar that has everything that yeast needs to grow except for a carbohydrate, and then we just add the carbohydrate, the one carbohydrate we're interested in. We put a bit of yeast on that agar and then we wait. If we see that there's nothing after waiting a little bit, then we just assume that it can't grow at all and we give it a 0. Whereas if you see this kind of really bold, opaque growth on the right, we assume it can use that carbohydrate and we give it a 1. These are the carbohydrates that we tested in ascending order of size from left to right. So we obviously tested nothing because we're good scientists and we use negative controls. We've got our little monosaccharides, some slightly bigger ones, and then our big inulin and wheat starch. We couldn't actually use bread fructans, that would be ideal, but they are very hard, I know now, to isolate from flour, and inulin is a good enough substitute for the purposes of the experiment. So this enormous graph is the growth scores of every yeast. So the growth scores are on the y-axis on the left, sorted by the starter that they came from up the top, and the growth carbohydrate on the right. And the only thing that I want you to see from this graph is that if you have multiple yeasts of the same species from the same starter, they more or less behave the same. So that tells us that we're probably dealing with the same yeast, just copies or clones of it. So if we reduce the noise and just look at one one species from each starter. On glucose and fructose, every yeast is happy as a clam. Regardless of whether it's a positive control or not, they are all just thrilled to be there. When you get to inulin, though, that's when things get a little bit more interesting. What we found, surprisingly, obviously the Cluveromyces, which makes inulinase, is going fine. The Kazakhstania is not as much, but the Kazakhstania boulderi in the red doesn't grow as well as the Kazakhstania humilis in the green. So now we also have to consider that maybe there's a species-specific property of inulin or fructan degradation that we have to consider, but we weren't expecting to find that, so that was really cool. And just to show you what I saw on the plates, the yeasts outlined in red are the boulderi, and you can hopefully see what I saw, which is that they definitely are not as happy as the others. There's so much more that we don't know about this genus. It's a very big genus, if you remember the family tree, and there's lots still to be looked at. Having something like Crohn's disease, I know from my big brother, is an illness with a significant burden on your quality of life. And it would be really cool if some of these sourdough yeasts that have been kind of under our noses for thousands of years, I think, it would be really cool if some of those were able to provide a solution. Thanks.
That's a hard act to follow, isn't it? Well done, Anna. Thank you. I just want to wrap up this presentation from people at the university with a few other considerations about sourdough microbiology, including the role of bacteria. So I love the idea of community, and I think GrAiNZ emulates all of those wonderful qualities of a real community that comes together. It's they talk, they speak, they're dynamic, and they work with one another. But this, of course, also happens in a sourdough starter. We wonder where yeasts come from a sourdough starter, and I've had several conversations over these past few days of where they come from. We don't always know, actually. They are likely to come from the hands of the baker, from the in-house microbiota, but also from the flour, potentially also water as well. But this slide demonstrates that you've got this daily feeding routines or even weekly feeding routines, however your baking regime works. You activate and you cultivate a dynamic collection of microbes with one another. And we've got some beautiful electron micrographs, a picture that I stole from our colleague Delphine Sicard, which shows you yeast and lactic acid bacteria growing in a with wheat starch in a sourdough starter there on the right. Anna's alluded to all of the different yeasts and yeasts that we've seen, and I wanted to introduce another level of this with a global study that we've published to collate all of those yeasts together. You'll see that on the donut here, the bigger colour of the donut is its abundance, and Saccharomyces cerevisiae is the major part there. But as Anna has told us, the Kazakhstania yeasts and the sort of greeny colours there are also important, along with a wide range of other yeasts as well. What I wanted to introduce into this conversation today was the role of bacteria. And a very difficult to read table up there in the top right-hand corner shows that there's approximately 50 or so species of bacteria that have also been isolated from sourdough starters. And so work that we're doing in the lab is to try and understand the interactions between these yeasts and bacteria together in the fermentation to understand how this impacts what we see in the loaves of bread that we love to eat and enjoy. I wanted to highlight and embarrass, I can see him laughing uncomfortably at the back, the sourdough starters that Chris Stafferton was kind enough to donate to science. You'll be familiar with Chris's work on making absolutely amazing gluten-free sourdough bread with his book Promise and Fulfillment, and I'm sorry I don't have the full title there, Chris, but you are welcome to approach him later to ask about this. Chris, this was pre-pandemic. He hand-delivered some starters that he has cultivated in his work using different gluten-free grains. So quinoa, or buckwheat, brown rice, millet, and teff. We wanted to know what was in there. So we, as Anna's alluded, we go ahead and we isolate them. This is one of Anna's slides again. And we see them in their colonies on a plate. And there we can identify what they are through looking at their DNA. You'll see a wide range of very complicated names. On the bottom row there, you'll see all of the bacteria. They're smaller than yeast. They are numerically dominant in a sourdough starter. So in an active starter, you might have about 100 to 1 cells of bacteria to yeast, but of course that dynamic changes during the fermentation. And of course, all of Anna's favourite Kazakhstania friends are indicated there as well. In the lab at the University of Melbourne, we've got a wide range of really exciting facilities to be able to understand this in a way. So we take Anna's isolates, other students in the lab's isolates, and we can cultivate those in a laboratory, so we're assured that they are a pure culture of that particular yeast or bacteria. Then we make dough out of them. We bake little pups. I think bakers have laughed at us for how cute and little our tiny little loaves are. But then we can present them to a sensory panel and say to the sensory panel, well, you know, is this different? Do you prefer the flavour of this or that? We can test to see the nutritional quality, the structural quality, the flavours of these different breads. And of course, you'll find people will flock to the corridor as soon as they smell the baking loaves come out of the oven in the hope to get a free little leftover piece of bread to take home. So Anna has talked about the use of yeast to make FODMAP-friendly bread, and I wanted to highlight some work that we've been doing looking at other species that we've isolated from Chris Stafferton's starters. Wickerhammeromyces anomalus, which is there on the left, and Pichia fermentans, which is there on the right. And we've been using a lactobacillus strain in partnership. We've done this work in teff and in buckwheat flour. A complicated slide to impress you all on how sciencey we can be. But what this slide shows us is very similar to what Anna showed. We have along the graph, we've got time of fermentation. So time of fermentation is incredibly important. And we have, by colour, the different dough treatments. So we have a wheat dough by itself on the top line there. We have teff dough in the middle and buckwheat at the bottom. And then as the fermentation progresses, we can see what happens to the total fructans during the fermentation. We have the dough by itself. We also have yeasts and bacteria together. And so you can see in some cases, and here I draw your attention to the top line, that by the end of fermentation, after 24 hours of fermentation, you can see that Wickerhammeromyces and lactobacillus dramatically reduce the amount of fructans that are present in bread. Of course, in teff and buckwheat are naturally low in FODMAPs to begin with, but still we see a reduction, particularly in the teff loaves, as the fermentation proceeds. So we have naturally an ecological system which can interact with one another to decrease the amount of fructans in bread to hopefully develop breads which are accessible financially, flavour-wise are fantastic, but are also nutritionally dense for people who can't otherwise eat them. So I'll wrap up this session, and we're hoping very much to have a vibrant Q&A in the end. We've found that Australian sourdough starters have diverse populations of yeasts and bacteria. The yeasts and bacteria that we find are similar to those isolates, so the same species that are found elsewhere in the world. we should see our sourdough starters as more than to just leaven and produce carbon dioxide. We should see our sourdough starters as a really a complex community that we need to cultivate and to encourage to be able to transform this raw dough into a fermented product that can be baked into bread, which is delicious and flavoursome and nutritionally dense. And we need to consider this as an ecosystem. So interactions between yeast and bacteria together can have more of an effect than one yeast alone. So diversity, as always, is key. Of course, the first acknowledgement is Chris, who generously donated his starters to science. The many people we've had through the lab who've done the work, and to you, thank you for listening. We would be very happy to take some questions here, and this can be to any of the three speakers. Rudi, would you be comfortable up here on a stool? Anna, maybe you'd like to come up as well, and we could use the microphone. Are there any questions we can ask? Jason?
That was fantastic, thank you. Just got a few questions about quality, how it's defined, and why this kind of work is not part of our quality definition. And what is the barrier to both having a genetic and processing response across our agriculture to having better, like, I guess having a FODMAP work? Like, is it just funding, or have we got a narrow definition, or what would grow?
Do you want to respond first, Rudi?
So you're talking about the quality of the grain or the flour that's milled?
Well, yeah. We'll start with the grain.
Yeah. So a lot of the grain in Australia's export goes for export, and the exporters are really, in my experience anyway, are keen on taking shortcuts, so they're not too keen on the quality testing. That's just has been my experience. It's been disappointing. So the quick and dirty tests are what's done. With the developments in agrobio, certainly we're capable of now doing more accurate quality testing, and it's really just to get to a point where, in the end, it costs, obviously, but I think there's a lot of exciting stuff happening where you can get fingerprints for your different material that will then let you say this belongs to this category, and particularly specialty wheats that are more suitable for what you're looking for. There's an incredible amount of material. There's on the health front in CSIRO, where I was for a lot of my life, there's wheats that are being produced that are high amylose, they're sort of like the maize equivalent, and they're just there. They're not being exploited. So it's from a invisible fibre sort of idea. Some of these are really valuable. So I'm not sure if that answers your question, but it's sort of, it's in the end it's costs, but there's a lot of things happening with spectral analyses that people are now starting through the DNA analyses, the NIR type analyses are starting to. I've always been a bit sceptical about them, but they're starting to being able to get more meaning from them as well.
What's the barrier to investment, though, from industry to make this happen?
I think I'll just speak up. Yeah. I think the exporters are a different world, and they dominate our grain, and so that then drives the breeding programs to produce certain things on a large scale, which then comes to people like yourself. But you're really you're not in control of what you get at the genetic level. That is, yeah. There's a lot of environmental things that can be done as well, but I found the grain exporters are quite. They work to a different the beat of a drum, yeah. So that it's the question. The answer to your question is that there are genuine. The business model for grain exporters really stop you from really driving a lot of this innovative research into defining your product carefully.
Okay. Go ahead.
In the back catalogue, there's a few weeks of the British cadenza and a few others like the spelt that have a higher ease of digestion, I suppose. Yeah. Is there particular markers that you have that you can search for in the database for
to match?
Yeah.
Yes. There are regions which are based on some, like the work that Anna's doing, which gives you some parameters to aim for. So the answer is yes, and the properties of the starch, for example, are one. The levels of there's a whole range of enzymes which are important in producing the starch, the amount of branching and all that sort of thing. So the answer is yes. The knowledge is there for defining both at the protein level, which is what I used to work with, and all the different products that are correlated with quality, and then also but the starch is the main component, and then there's a lot of the minor component-wise, like the fructans and the arabinoxylans, which are involved in water absorption, which, again, the technologies are now there to provide you the diagnostic, which is becoming cheaper. And I think, as I say, I'm now in agribio part of my time, and it's very exciting to see that research level of the Department of Ag here in Victoria.
Sorry, I have two very quick questions. With the sourdough starter, the bacteria and yeasts, do you see or expect the populations to change over time, or is it a fairly constant? And the second half is, with the fermentation time, is temperature a factor in your...
Maybe you should talk about your honours work, about how things are stable over time.
Oh, yeah.
Yeah.
The answer to your first question is... sure, yeah. So the first question was about whether the kind of relationships between sourdough yeasts and bacteria change over time, if I got that right. And the second was about, we talked about time of fermentation being a factor with fructan content, and the question was whether temperature is as well. So for the first, the answer is that sourdough ecosystems tend to stabilise over time, and there are actually quite a lot of studies looking at, I think that the term they usually use is the succession of, they talk about waves of, we have, this stage dominated by this bacterium and this yeast, but which yeast and which bacterium and how much and whatever, that depends on the individual circumstances, but they do tend to find their own set point. And I spent time in honours every day coming into the lab. I made a kind of an artificial synthetic ecosystem in little plates that I subcultured or backslopped every day. And yet I found that the numbers of yeasts and bacteria tended to level out over time, and they tended to find their groove partnership-wise very quickly. They tended to just, yeah. The second one, about temperature being a factor, the answer is probably. I haven't read anything directly about it, but I can't imagine it not being a factor. I'll pass over to Kate.
Yeah. A microbiologist might say that every microbe has got its optimal growth temperature, and so that is set by the conditions where you maintain your starter. You would be unknowingly selecting for microbes that grow particularly well at whatever happens in your bakery or in your home or whatever it is. And so over time, even if the ambient temperature goes up and down a little bit, you'll be selecting for communities that can deal with that and can maintain that balance. Something we haven't spoken about today is that yeast and bacteria need one another. They need one another to coexist during this fermentation. Yeasts are not very good at breaking down carbohydrates, generally speaking. They're very good at taking up sugars, so simple sugars, as Anna's seen. But they're very good at letting go of nitrogen. So they'll produce the amino acids that they need, but others will just overflow out of their cell. Bacteria, on the other hand, are something called fastidious. As Gabrielle was talking this morning, they're the fussy eaters. They can't make all of their amino acids, so they get them from the yeasts. In return, bacteria tend to have a wider range of enzymes that they can secrete to break down carbohydrate, which the yeasts use. So you get this sort of circular dependency. So they do need one another, and so regardless of temperature, you would be selecting for them in this balance. I'm sure they will all be aware of cases where your sourdough, you don't get a proper leaven or rising when it's very humid or it's very hot or whatever, but generally speaking, you've selected for it unknowingly.
I just want to have mic privileges for 1 second. I just want to apologise to Chris Stafferton for falsely claiming that we made some of your sourdoughs. I was subtly corrected from Kate's slide. Sorry. Not all of them, some of them were us, but most of them were Chris Stafferton, so credit goes to him.
And now Chris gets a question.
Okay, question. Similar to Simon's question, but just a spin on that. The communities that establish of yeast and bacteria, if they establish on one substrate and you change the substrate, is that going to move the makeup of the community?
Maybe is the answer. The answer is I don't really know, but what we do have is observational. I was pretty sure when I started out that the same starters in the same bakery would have the same communities in them. But that isn't true, as we know from your starters, Chris, but also from the Tivoli Road starter, which was one of the first starters I looked at. And Anna, I was reflecting the map of where we get our starters from is basically where I go on holidays. And I introduce myself to bakers and ask if we can look at their sourdoughs. Tivoli Road... Soon. So Tivoli Road, when Michael and Pippa were running, it was one of the first starters that I looked at, and we took a sample of a wheat-based starter and a rye-based starter, and the bakers were busy and it was all a bit uncomfortable, and they gave us this enormous package of it and was like, oh, we only need like 1 gram, and it was, a bit confusing. But what we found is that there were different communities in the wheat starter than there were in the rye starter. And I thought that was unexpected. I would have thought that it's just, a baker's hands mixing up this one, they might also mix up that one, and there would be exchange. But because of that and because of your starters, Chris, we know that that's probably not true, and that the wheat or the flour that we're using selects for the community that is established. Just on the left there, the hand there.
I've got 3 questions. Can you identify yeast under a microscope? And if not, can we send our starters to you to be analysed?
Please do.
And the Kazakhstania genus that you're interested in.
Sorry, I think that's me.
Would that be able to just be used in isolation in a home sourdough starter, or would airborne yeasts and other things get into that community?
Maybe I'll repeat the question. So the first question was, can we identify yeasts under a microscope and can they be sent to us? And the answer is kind of for the first one. We tend to take a little bit of their DNA and sequence that, and we get a more accurate read on what they are, just because maybe you noticed on the slide that Kate had with the photos of the yeast is that they often look quite alike. So identifying them by what they look like is not always the best way to go. But yes, please send them to us. We make the map bigger. With the Kazakhstania humilis, it's, I think after Saccharomyces cerevisiae, it's the most common yeast in sourdoughs. Was your question whether if you bought a pure culture of it, you could use it, or would it be the only...
For FODMAP
right.
purposes, you know, could you grow it at home, and would it become, does it need, like, because you were just saying how bacteria and yeast really need each other, and I imagine the whole community is all, they're all doing bits. So, how do you keep that isolated? You wouldn't, and that you probably wouldn't want to keep it isolated. Kazakhstania humilis kind of has like a tiny best friend, and it's one of the lactic acid bacteria called, it's named after San Francisco. It's Fructolactobacillus sanfranzisensis. And they have this like really, it's actually, I mean, I don't know if you would call it famous, but among sourdough research, it's a well-known co-occurrence. So usually where you find Kazakhstania humilis, you usually find the bacterium as well. And you wouldn't really want to split them up because, as Kate said, they need each other. And actually, it's good that you mentioned that, because the work that Emily mentioned, I'm going to Delphine Sicard's lab. I'm so excited. The work that I'm doing there is looking at the partnership between Kazakhstania humilis and its little friend to see whether, I just don't want to say the name again, it's too long, to see whether, to see what the mechanism of their partnership is. In terms of the other yeasts that are in the community, you have to keep the Kazakhstania kind of the most abundant in order for it to be kind of FODMAP useful, if that makes sense.
It would be, I think that would only happen if a yeast company was prepared to make a single culture of Kazakhstania and sell it, which some of them seem to be ready to approach, because everyone's looking to find a niche yeast bacteria in humans and companies as well. And so there is some interest in producing interesting yeast commercially so that we can keep it sequestered, if you like, and just working. It would be extraordinarily difficult to do that in a home or bakery, because anytime there's something fermented, you'll see it yourself. You see the vinegar flies come in, the vinegar flies sit down and eat. They've got yeasts in their gut. They leave yeast there, they take them with them. You know, you wouldn't be able to keep a pure culture in that way. Is that finished? Okay. Thank you. I think we've got it. Oh, one more.
I was going to ask a question. Now that the wheat breeding has been privatised in Australia, and we now get Roundup ready varieties that farmers get to use, and they also have farm-back royalties that they have to pay to the breeders of those wheats, is there any attention being paid to actually breeding varieties that are more sort of beneficial for organic growing practices and flavour rather than just pure volume of yield?
Yield is still the big driver, but it's been terrific to see Intergrain in Perth actually have made quite a big investment with AgriBio here in Victoria to produce the DNA sequence-based analyses of all their varieties. And so that's a start to try and deal with your challenge. And if there are particular features of the grain or of the genetic material that's advantageous for organic growing, then they could select it. And if there's a big market, a specialty market.
Is there incentive for that to be happening in the breeding industry, or is it still more focused purely on volume and production?
This is part of this business model I mentioned before with the export grains. But I'm finding the group in Perth particularly good in sort of making those investments which are sort of publicly available. The AGT in Adelaide is a bit more sort of inward looking and they don't do that sort of investment, but certainly I think the will is there, but the driver is sort of, the business models just aren't there at the moment. You know.
Thanks, Rudi. All right, everybody, join me in thanking very much these fantastic scientists.